Okay, hello everyone. We'll be going through chapter 2 now. Um, so this is about describing data. And one important way we share our results in biology as well as in many other sciences is to uh, use graphs to describe the results, to summarize and describe the results, in addition to doing estimation and hypothesis testing that we'll be learning about later. And why are graphs such a fundamental piece of statistics, of data analysis? Well, humans are visual species, just like other primates, and we can see patterns in data visually uh, relatively easily. It's an efficient way to convey information, to explain things. And when we present our results, we use it also for um, preliminary analysis of our data. So once we collect data to understand what's going on, to identify main patterns, to identify potential problems in the data, it's always helpful to plot the data first. So looking at the data as numbers usually doesn't tell us much, but when we plot it, we can identify what kind of main effects are there, what kind of factors influence the data. We can see that very efficiently, so we can get a feeling through graphs. Yeah, and as we mentioned, so both to to um, digest and get a feeling about our data once we've collected it, and to decide how to analyze it, we need graphs, and also to describe our results, we need graphs. And this is actually from one of my earlier works. So different set graphs, as you see, describing gene expression changes in the brain in humans and chimps and other species, both with age, and so how gene expression is influenced by age and species factors. So different graphs telling us about different stuff. And this was that was kind of stone age of uh, graphic transcriptome descriptions, and this is a much more recent study of single cell transcriptomes, this time from the testis. And here there's also they also have a cartoon, as you see, that describes how sperm maturation takes place, and then description of real transcriptome data, RNA sequencing data that describes expression levels of single cells measured in single cells. Now that changes over time, nice representations as you see. Uh, yeah, so graphs contain a lot of information that help us understand what's going on. Um, we will not be, well, we'll be partly talking about some types of these graphs, but we'll start from the very basics, and we'll start with bar plots or bar graphs. And these are graphs to describe frequencies or relative frequencies for categorical data. Frequency we already learned last week, so the number of occurrences uh, of a certain type of categorical variable in a sample. That's the frequency, and relative frequencies are just scaled versions of frequencies. So if we divide the frequency of each type of observation with the total number of observations with the sample size, we get a proportion or a fraction, and that's relative frequency. And the example in the book is with different cause of death in U.S. teenagers. And here the sample size, as you see, is 13,000. Um, um, and we have teenagers, cause of death collected from teenagers in a certain number of years. Oh, sorry, in just 1999, 1999. And so this we can see as a sample that might give us some information about uh, the population here. The population would be cause of death across um, teenagers in different years, not just 1999, but also other years. Or we could also, the population might also be described as perhaps cause of death uh, in teenagers across the world, but then that might be then this one might be considered a biased sample, right? Because cause of death for American teenagers are not necessarily representative of the whole world because that's a rich country. Okay, so we have accidents and homicide, so killings as the first two, and then suicide, tumor as the fourth case, etc. So what's the variable here, and what kind of variable are we talking about? The variable itself, as you see, is a categorical variable. It is nominal or ordinal. It's nominal, right? There's no order, inherent order among these different types, and we have data on how frequent each one is observed. And we can describe this result as a bar plot. So instead of showing it as a table, we can show it as a bar plot, which, as you see, it conveys the information that how dominant accidents are among all deaths. This is conveyed pretty efficiently in this graph. Just looking at the numbers, it's perhaps more hard to see. Uh, this gives a better feeling. Um, and indeed, accidents, you might already know that in teenage years, the brain is working somewhat differently, and risk-taking behavior, it's been observed, is much stronger. That seems to be some kind of universal pattern, at least as far as I know. This is popular science, and that could potentially explain why accidents plus homicide and suicide are relatively frequent among teenage deaths. But of course, to put this in context, we would also need the cause of death for other ages as well, um, and from other societies as well, of course. Okay, one small thing: could the the shape of this graph, would it be different if we had instead of the number of absolute number of cases, if we had plotted relative frequencies? The shape wouldn't be different, right? The y-axis would change, but the shape wouldn't be different. So rules to make a bar plot: if the data is ordinal, if there's order in the types of categorical variable, then the bars should follow the variable's order. But if there's no order, if the variable is nominal, then you may order by frequency to aid in understanding the patterns, as you see in this case. Um, and then there are gaps between that's a characteristic of a bar plot, a tradition. uh, the bars are not drawn next to each other, but with gaps to emphasize that these are categorical variables. And it's also helpful here to add extra information in the legend. Here, for instance, it's described. First of all, we learn about how many variables we have in total, 10, and then what this is all about, and also the total number of cases, the total frequency. That's also important to describe. So always in the legend, try to provide as much information as possible to the reader to make it clear what. So the rule should be when the reader looks at a graph, he or she should understand what's going on without having to look into the text. Okay, that will be a good graph. Um, some other types of bar plots, which now in this case it's like in a sense a 2D bar plot where we have two types of variable in fact plotted together. As you see here, the y-axis is relative frequency anyway, uh, but most important is we have also men and women, so gender as one categorical variable, and some response to some question as the other categorical variable, and the two are plotted together. And we can again easily see something we might not really see so efficiently. Just look, we would not have seen just looking at the numbers. We can see very clearly looking at the data that there is an increasing discrepancy in the responses between men and women, from going from a strongly agree to a strongly disagree. So that is, see, that can be easily seen here. Um, and yet another case. So this is uh, an ordin sorry, a nominal categorical variable, again, frequencies, count frequency, same thing uh, being described. And here there hasn't, um, frequencies, sorry, the category types have not been ordered according to their frequency, as we see. But that's okay. It would, of course, still be easier if it had been ordered like this, easier for the reader. Okay, and now we turn our attention to another similar type of graphic, but has some fundamental difference. So what we had been talking about were bar plots that describe frequencies of categorical variables. Histograms describe frequencies of continuous variables, okay, of numeric variables. And the biggest, most clear distinction between the two types uh, in terms of visualization is that here the bars have gaps in between to emphasize that we're talking about categorical variables, whereas in histograms you have no gaps. Okay, and you should know this, and this very traditional way of describing two different types of variable, and yeah, you should know. Um, so histograms are graphs that describe frequencies, relative frequencies, so frequencies of numerical data. And here we bin the data in windows, as you see. So this is one window, or one bin. Bin is more commonly used. Another bin, another bin. And then we plot the frequencies of observations that have values within that bin. And there is, yeah, no gap between bars indicating continuity. So let's first look at this table again from the book. So we have species abundances of, I don't know how many species of bird, how many, the table describes how many times each species have been observed across 4 surveys in some region in North America. So the great roadrunner, as you see, has been observed only once. The bronze cowbird also only once, while the dove has been observed 600 plus times. Okay, so the variable here can be described as the species itself. And then we could describe this plot as a bar plot. We can instead also describe the variable. We can deal with this data by treating the variable as species abundance, not the species itself. So uh, we might be not interested in the species per se, but we might be interested in overall abundance of species. So might be interested in the question: do there exist many species with which are very frequent, or do there exist many species which are very infrequent relatively? And we can try to address this question whether uh, common species are or frequently observed species are also frequent, or frequently observed species are infrequent, how the distribution is of this type of abundance data using a histogram. So coming back to my question, so what variable we are dealing with here? If we're interested in describing abundance of each bird species, if we were interested in bird species itself, then our variable would be bird species and its frequency, and then we would have a bar plot. But if we're interested in the question about bird species abundance in general, then we can describe the same data in a histogram. And to make a histogram, we may need to create bins, as we mentioned. So here, the data ranges from 1 to 600 plus. So we can have 13 bins going by 50. And then we would have, for instance, all these species until here. Um, I don't know how many, 20 or something. All these species in the first bin. So species that are observed between 1 and 50 times, and then species that are observed 50 to 100 times, just one observation. Sorry, 50 to 100 here, right? 4 observations, and then 100, 101 to 153 observations, and so on. So this is a summarized version of the same data, summarized based on abundance. Now our variable is abundance, as you see, not species itself. And we have 28 species that show abundance between 0 and 50. You see the point here, where the species has been observed only once to 50 times, and then 50 to 100, etc. 50 to 100 only 4 times, etc. And if we plot the values here, we have a histogram that looks like this. In the legend, as you see, the total number of species is also described. The legends tell us what's going on. This is the frequency, number of species. This is the abundance per species. How many times? Sorry, the abundance, uh, yes, per species, how many times the species has been observed in this national monument park something uh, during 4 surveys. So 28 species have been observed between 0 to 50 times, and then 4 species have been observed 50 to 100 times, etc. And then we have one kind of outlier species. So the shape itself tells us something about the data distribution, what kind of process is going on. It, if it's a normal distribution, that tells us something. If it's an exponential distribution, which would fit this one more, it would tell us something else. Um, it Plotting the histogram also tells us about the existence of outliers, as we see here. Um, so here, let's look at a couple of different types of histogram, and let's learn about the names people use to describe such distribution. So uniform distribution is a type of distribution where different values within the range occur at equal frequency, at more or less equal frequency, or equal probability. Um, so this is a uniform distribution, and this is a bell-shaped distribution, a normal type. Normal is one type of bell-shaped distribution. We'll learn about others as well. Um, Asymmetric distribution This is unimodal, so it has a single mode, uh, but it has a tail. It's not symmetric, as you see, a tail towards the left-hand side. We would call this a left tail distribution. Skewed is another way. Skew is a commonly used term to describe that a distribution has a tail. Left skewed would mean a tail to the left towards lower values. Right skewed would mean a tail to the right side. So this would be a skewed distribution, skewed to the right with a right tail. Um, and a bimodal distribution has two modes, so two peaks. A mode is a peak, uh, and these are unimodal distributions. This basically doesn't have a mode, and this is a bimodal distribution. If you see this kind of pattern, it suggests there are two underlying populations within that sample, or the sample is obtained from a population which has two main distinct types within it. Okay. Yeah, so distributions can be of different type. We'll learn some of them, uniform and normal. We will learn exponential and gamma. We won't learn, but there are many types, which are also described in mathematical functions that, as we had touched upon in the previous chapter. Uh, Mode of a distribution is the peak, is the most frequent bin. So this is the mode, but here we have two peaks. So after a value, we have another peak. And so that's why we call it bimodal. Whether a second peak exists or not, whether it's just noise, that's something that people have developed tools to identify. Um, and of course, there might be even more modes other than two. Skew is asymmetry, as we mentioned. We're talking about tail and outliers. This is a commonly used term. You should definitely know. These are extreme observations that seem to be under the influence of some other factors that do not uh, possibly influence the rest of the observations. So some kind of extreme situation. For instance, in a transcriptome experiment, an outlier might be caused by a technical problem. So if RNA was degraded when you were collecting material from a sample, that might be reflected as an outlier observation in a transcriptome description. So in the transcriptome plot, this is a transcriptome plot, and if we had an individual which was over here, we might consider it which was a human or chimpanzee, but didn't match any other human or chimpanzee. This we might call an outlier. Okay. Rules about histograms. So choosing the correct number of bins is something important, and we'll see how choosing bins too few or too many might help conceal signals, or yeah, it might cause the reader to miss signals. So this is a set of data, the same data represented in histograms. The only difference is how the bins have been constructed. Here, bins go at steps of 0.5, as you see. Here, bins go at steps of 0.1. And here we seem to observe a second peak, right? There's a value and then an increase in frequency. That might be, I mean, we won't study them here, but there are, again, methods that can see if the second peak might be non-random or not. This, assuming this is real, this graph still represents that second peak, whereas in this graph, basically, due to how wide the peak sizes have been drawn. In this graph, we don't see the second peak anymore. After all, we just see a single one. So this is something to keep in mind. Um, yeah, and one way, there's no straightforward simple rule to decide on bin size. So the best is to plot your data at in different ways to make sure that you're not missing something or you're not over interpreting. Another rule about histograms that you should know. I mean, in real life, you won't be most likely drawing histograms manually, but you should know what the algorithms that draw them for you, like in R or in Excel, what the algorithms are actually doing. What they do is, as you see here, when we have, perhaps here, when we have boundaries that overlap. So here 0 to 50, 50 to 100. If we had exactly 50, what to do? Which bin we should include it in? And the rule is to add those observations to the right-hand bin, so to the higher bin. So in the case of 0 to 50, 50 to 100, if we had an observation of with value 50, we would have included it in the bin that goes from 50 to 100. Likewise, from 250, if we have a 250 observation of value 250, we add it, and we have bins of 50, we add it in the 250, 300 bin, not in the previous bin. Another one point to keep in mind is to prefer 0 as baseline, as in here, not move the baseline above 0. Having bins that break at readable numbers, at round numbers, is helpful usually. Otherwise, I mean, it doesn't really matter, but it's just a issue of convention of you know, making nice looking graphs and not confusing your readers too much. It's just easier. The easier a graph is to understand, to grasp, to see, and to grasp as fast as possible, the better. And then always add information about what's going on, what variable we're observing, and the total number of observations in the legend. Okay, so this is, and always, always, always add X and Y axis labels. Okay, so again, when the reader looks at the table, he or she should understand what is being displayed. Shouldn't have to go and read the main text. Okay, those were histograms and bar plots. Now we will be talking about some other description of frequencies, slightly more confusing than histograms themselves. They're, yes, another representation of numerical data, including frequency data, and they can be used for when we're trying to describe frequencies of multiple different distributions. You'll see examples in a second. And the y-axis here, they show percentiles or quantiles. Percentile is what percent of the data have a certain value. That's the percentile. If, for instance, a value x is the 5th percentile, which also is the same thing as 0.05th quantile, then this means 5% of all values in our sample have a value x or lower. So 50th percentile, or the 0.5th quantile, same thing, is the value where half of the sample has values lower than that. So that's also called the median, in Turkish ortanca. Okay, so the value in the middle of the sample. If we sort the values the value in the middle is the median, also the fifth percentile or 0.5th quantile. They all refer to the same thing. Okay, so the cumulative relative frequency distributions, they describe a graph of quantiles or percentiles, not frequencies directly. And the y-axis, it might represent percentiles or quantiles that go from 0 to 1 or 0 to 100. And let's look here. So here we have percentiles that go from 0 to 100. And here on the x-axis, we have the variable itself, which is a change in precipitation. So this has been measured. Apparently, it has values going from about -20 to more than 120. Some kind of change observed somewhere, and it can be, apparently it can have positive values or negative values, and the underlying, so the underlying distribution is such that, so we can see that the values start from this point, something like -15, and then about 10%, here what we can see is 10% of the values have a value of 6% or lower. 6% is change in precipitation. Let's call it just 6 for simplicity. So change in precipitation value 6 is observed uh, in 10%, in less than, or oh, sorry, in 10% of the sample, and 26 is the median value. So half of the observations have a value 26 or lower, and then 90% of the observations have a value 57 or lower, as you see here, and all of the maximum observation seems to be something like 120, 130 perhaps. So this is a description of the original data, which has been converted into a cumulative relative frequency plot. The original data would be, we can predict it would be something like exponential. So it would be something like this, where the bulk of the data would have low values. Anyway, we can also see that the median is something like 26, somewhere here. And in cumulative frequency plots, the more the stronger the slope, the higher the slope, in that region we will have more of the data. Okay, wherever there's a lot of data, that will create a strong slope. So probability of or the relative frequency changing fast. Okay, so the same data that we saw here, we can describe also in a cumulative frequency plot. How would it look like? So the data that gave rise to the cystogram, how would it look like in a cumulative frequency plot? It will look like this. Okay, again, big part of the, so this rapid, this very strong slope means big part of the data is already here, and indeed already within this small value of 10 or something, we have half the data. So this is the median value, right? This is the median. You can also put it to median value at 0.5, and then here we have about 75% of the values, and then it slows down, and then finally we have the maximum value reaching 1. Yeah, so these two are describing the same pattern, but in different from different dimensions in a sense. As you see again, in a cumulative relative frequency plot, the higher the slope, the more data in that range. The lower the slope, as in here, less data in that range. So most of species have high frequency. Uh, sorry. No, just the opposite. Uh, Many species have low abundance here. So the most of the data is concentrated here, uh, with low that they are low abundance observed relatively rare, and few species are observed uh, at very high abundance, and that is described, that we can see from this low slope. If we had a distribution that looked like this, where bulk of the data had high values, then this one would go would have a shape, the cumulative relative frequency plot would have a shape just the opposite. Okay, so low slope and then high slope. Okay, and cumulative relative frequency plots, they are, uh, to admit, they're a bit more difficult to digest and to understand the patterns, but they can be helpful to describe multiple frequencies, and they're pretty commonly used, not as frequent as histograms, but pretty commonly. And so it's useful that you understand how they are interpreted, that you can interpret them. Um, Box plots we will talk about somewhat later, but yeah, so let me, if I could give a break here, and we will continue in the second video.